Welcome! In this video, we'll review key concepts in DNA replication through a series of quick questions. When each question appears, pause the video, try to answer it yourself, and then resume to check your understanding. Question 1. A picture of a single origin of replication in a eukaryotic cell is shown. With respect to the vertical line, in which direction does replication proceed? Left, right, both left and right. Answer: both left and right. Replication is bidirectional, so two forks move outwards in opposite directions from the origin. Question 2. Shown here is an origin of replication with two forks. Consider sites A and B relative to fork 1 and C and D relative to fork 2. Which template strands are replicated continuously? A template 1, B template 2, C template 3, or D template 4. You may select one or more answers. The answer is A and D. DNA polymerase synthesizes 5' to 3', so the leading strand at each fork is the one whose template runs 3' to 5' into the fork. In this diagram, those are template 1 at site A and template 4 at site D. Question 3. Using the same diagram as question 2, to which sites can the primer 5' GUUCC 3' bind to initiate replication? Answer: the primer will base pair with a complementary antiparallel sequence, which is 3' CAAGG 5'. This sequence is found on template 2 and template 3, so the primer binds at sites B and C. Question 4. Shown here is a picture of a single origin of replication. On the left side of the vertical line, which template strand, top or bottom, will be more affected if DNA ligase is only partially functional? Explain why. The bottom template strand. On the left fork, the new strand built on the bottom template is made discontinuously. As Helicase opens the fork, the bottom template is exposed in short stretches rather than all at once. Because DNA polymerase can only extend DNA in the 5' to 3' direction away from the fork, it must build the new strand in many small Okazaki fragments each time a bit more template is revealed. Each fragment then needs to be joined by DNA ligase. If ligase is only partially functional, these fragments cannot be efficiently sealed into a continuous strand, so replication of the lagging strand from the bottom template is most affected. Question 5. You are examining an E. coli mutant whose DNA polymerase has an error rate of 10 to the power of -3 instead of the normal 10 to the power of -6. Which activity is likely missing? Select all that apply. A. 5' to 3' polymerase, B. 3' to 5' exonuclease, C. 5' to 3' exonuclease, D. 3' to 5' polymerase. Answer: B. 3' to 5' exonuclease. This proofreading activity allows DNA polymerase to remove incorrectly paired nucleotides from the growing end. Without it, the error rate increases dramatically. Question 6. In prokaryotic circular chromosomes, DNA replication termination occurs when A. Replication forks randomly collide anywhere along the chromosome, B. Replication forks meet at specific TER sites and thus proteins block further helicase movement, C. DNA polymerase reaches the last gene on the chromosome, D. RNA primers are removed. The answer is B, because the question specifies prokaryotic E. coli genome, which has one origin of replication. Choice A would apply to eukaryotic linear chromosomes, which have multiple origins. Question 7. During DNA replication, linear chromosomes face a special challenge on the lagging strand. Which statement is true? A. Telomeres act as buffers so that DNA lost during lagging strand synthesis does not affect essential genes. B. Telomerase repairs internal DNA gaps. C. DNA polymerase can fully replicate chromosome ends without help. D. Telomerase prevents shortening of the leading strand by adding nucleotides to RNA primers. The answer is A. Telomeres are long repetitive non-coding sequences that prevent essential genes from being eroded. B is incorrect. Telomerase does not repair internal DNA damage. C is incorrect. DNA polymerase cannot fully replicate chromosome ends on its own. D is incorrect. Telomerase extends the 3' end of the template strand, enabling lagging strand completion. Question 8. A researcher inhibits glucose metabolism and finds that both de novo and salvage nucleotide synthesis slows down. Which step is most directly affected? A. Formation of ribose 5-phosphate via the pentose phosphate pathway. B. Synthesis of aspartate and glutamine. C. Reduction of nucleotide diphosphates to deoxynucleotide diphosphates. D. Reattachment of purine bases in the salvage pathway. Answer A. Both pathways require PRPP made from ribose 5-phosphate. If glucose cannot enter the pentose phosphate pathway, neither pathway gets the sugar backbone needed to make nucleotides. Question 9. Helicase and topoisomerases differ because A. Both unwind the helix, but only topoisomerase breaks hydrogen bonds. B. Helicase unwinds the helix. Topoisomerases relieve torsional strain ahead of the fork. C. Topoisomerase opens the helix. Helicase seals nicks. D. Helicase adds primers. Topoisomerase joins Okazaki fragments. Answer B. Helicase separates the strands, while topoisomerase cuts and rejoins DNA to relieve supercoiling. Question 10. During DNA metabolism, different processes start at specific sequences. Where does DNA replication begin? A. A promoter. B. The start codon. C. A chromosomal site where helicase and replication factors assemble. D. A splice junction. Answer C. Replication begins at an origin where helicase and other proteins load onto DNA. A is for transcription, B is for translation, D is for RNA splicing. I hope this question and answer tutorial helped reinforce the key ideas behind DNA replication. If you found it useful, give the video a like and subscribe for more quick molecular biology reviews. Thank you.